we're victims of our own success, in a way. That the genetics revolution, started by Jim and Francis and others, has just made advances in leaps and bounds since 1953. We can do now what would have seemed like science fiction back then, or even 20 or 30 years ago. It continues to increase. But the techniques that we use to study the biochemistry and the cell biology of cancer, which is my field, are very similar to those used in 1953. It's a difference in the way the technologies have advanced. So that's one factor. And the second factor, I think, relates to something Helga said this morning about promises. We overpromise. I review a lot of papers on cancer, and I can assure you that almost all of them end their abstract with saying, this information will yield a cure for cancer, or something like that. And it doesn't. Now, that information can be very useful, and maybe in 20, 30, 40 years it will turn out to be useful. But I think that we as scientists, this is our fault. Perhaps we're doing it because we're all competing with funding, for funding. But I believe we overpromise.
That last sentence is something new that began probably 10 or 15 years ago. It used to not be in papers. It would be interesting to know how that came in. In fact, in many years ago, when we would all write in our papers, this is a novel protein or a novel mechanism, the editors would strike out the word novel. They should probably strike out the last sentence that promises a therapy.
Right. Yeah, Neil, you've spent a lot of time making knockout mice and gene-targeted mice. Have you been barking up the wrong tree?
Well, I mean, I don't think so, but I mean, so just to comment on this, why progress isn't quicker. I mean, we heard this morning that the sequencing power has increased 1 million times in a decade. And just before this session, I met a student who worked with us in 2004, Thomas Linder, and he identified 4 genes that we felt were important for regulation of mitochondrial function. Now it's 8 years later. We have knocked out all of them in mouse. We have overexpressed them. We and others have crystallized 3 of the proteins. We have done lots of mechanistic biochemical work. And we feel that one of these proteins, we know in great molecular detail what it's doing. The other 2, we have good hints what they are doing. The 4th one, we still don't know. So, I mean, mechanistic work, I think, is just very difficult and much still very slow. So I think this is also hampering progress.
And is that acceptable? Is that acceptable? So we say mechanistic work is very slow, and it is very slow. What's happened over the last 50 years is some things have gotten very much faster. And as you say, some incredibly faster. We did, as a scientific community, work really hard to make them get faster. A lot of attention has gone to structural genomics, let's say. Much less to trying to do the same thing with functional studies. I think while I completely believe in the fact that people have to do individual studies on mechanism, I think we underinvest in the tools that could dramatically accelerate our understanding of physiological circuitry, of cellular circuitry. And I think, we can agonize about all that, or we can say, look, we've been very successful at each of the steps. The next step is we have to bring function to the same level that we've brought these other things. And then, frankly, we have to do the same thing to therapeutic development. If you ask about what is even slower, the actual development.
Fellows don't really want to do biochemistry and physiology. They're seduced by all the successes as you and your colleagues have been able to create in developing new technologies. And that, as a result, is very hard. In our lab for the last 40 years, and the first 20, so there were 2 or 3 people that always worked in cold rooms. Now we are lucky to keep 1 person in the cold room. In other words, that shows you that shows you the disappearance. But even more surprising is what's happening in a medical school curricula where biochemistry and physiology are almost disappearing. In fact, my colleague Mike Brown visited Johns Hopkins last week, and he was told that biochemistry has been sort of reduced to 5 days in the freshman course. And that was by members of the biochemistry department. So, and that's going on all over the country, is because one tries to get all the basic sciences in a very restricted time period so one can begin to teach translational medicine. And you see the same thing at the level of the NIH, where much money has been siphoned away from what would be basic fundamental work at the intramural as well as extramural that's being put into large projects. And I think that we, in the long run, are going to suffer, because if you look at the 10 most important drugs on the market today, every one of them required 10 to 20 to 30 years of biochemistry and physiology before they ever got there. And I think that's, I'm talking about a really important drug when I say one on the top 10, not a minor drug. And I think that really is telling us something.
Very clear.
I'd like to pick up on Joe's point. It seems to me that when we're teaching medical students or PhD doctoral students, that the most important thing to teach them is how to have an open mind about the possibility of being creative when the next wave of discoveries comes along. That it's not necessary to know every detail of how to run the most recent Illumina instrument. You can learn it, even I can learn it, but it's not going to serve them well in 5 years. What's going to serve them well in 5 years is an understanding of a genetic way of thinking, of a biochemistry way of thinking like a cell, of a feeling for the organism. And it worries me, speaking as a translational scientist, it worries me that if we focus on translational research to the exclusion of basic research and the creative process of making a hypothesis and testing it, using whatever tools you need, that our students they won't be able to pull out of the air the hypotheses for the next generation of experiments. It's very much what you say about a concern that we are teaching only to the result rather than to the process of doing science.
So,
just to add a couple of comments, maybe they've already been touched on in a way. I see that with any kind of process, you have a rate-limiting step, and drug development seems to be the rate-limiting step here that leads to the big problem. And as Joe has said, mechanistic understanding lags, and one often can have a terrible drug target by present-day standards, and it's just impossible to do anything with it. And yet, if you understand the process in its totality, you might have several targets to select from. And one can work around the drug development issue, at least for a time. But what I see in general is that what needs to be worked on, what needs to be improved, is very clear. And there has to be some really radical advance in the way we design drugs. And certainly mechanistic understanding is very fast compared to the way it used to be, but it too can be improved in certain crucial areas. And the poor training in biochemistry, that's one of the problems, but there are others that are just built into the system.
So 5 days is not enough?
Certainly not.
So, sounds like we can perhaps summarize that the bottleneck is downstream of genomics. It's more a problem of the time it takes in mechanistic research, the methods we have. Eric is advising those of us who work on disease models and similar to refine our models and learn from the genomics people. And then, as Mary Claire said, if we want to do translational medicine, we have to have something to translate. So this is for the decision makers, that you cannot just use translation as a magic word. You have to have fundamental research and the knowledge that comes out of that in order to build the translation and achieve the results in medicine and other areas that you would like to accomplish.
You cannot translate the Bible if you don't speak Aramaic. you cannot translate Shakespeare if you don't speak English. You have to understand the fundamentals of what it is you want to translate.
So we also need better language education in our science courses and the medical schools.
I think you are doing very well in your country in that way.
Right. Let me switch a little bit and ask you about gene-environment interactions. We've been very much focused on genes for obvious reasons, and that's the whole topic of this meeting. But for many diseases, cancer being one and heart disease being another, gene-environment interactions play a major role, and environmental factors come out very strongly in all statistical and epidemiological studies of disease. How far are we from understanding the gene-environment interactions? Do we know enough? Or if not, what can we do to move ahead here? Bruce, would you like to start?
Well,
sorry. Yeah, okay. Yeah.
Okay. It depends exactly what you're talking about with gene-environment interaction. Generally, we're thinking of a selective pressure in the environment. And probably there's nothing more stringent as a selective pressure than infectious disease, at least at present. It's something that kills about a quarter of all people who are born on the planet, kills most of them at a young age, and definitely it has a huge effect on the shape of the genome overall. And we're just beginning to understand it. We can point to a few isolated examples where mutations have been driven to very high frequency, even though they're deleterious. We know of some cases where there are individuals who are highly resistant to lethal infectious diseases at relatively little cost, and these all give hints of what we might do therapeutically in the future. But there's a huge amount left to be understood. If we were to sit and make a list of all the genes that have non-redundant function in combating infection totally across the board, we might come up with about 300 or 400, but there's a suspicion that it's in the thousands, the numbers that are really important.
Bert?
Yeah, I think that's a very important question, and science in general, particularly genetics, has really taught us a lot about this age-old question of nature versus nurture. And the answers, I think, are becoming clear, and perhaps the best way to describe them is to cite a study that was done in 2000 by people in Sweden, Liechtenstein and Amici, published a paper in New England Journal in 2000, where they looked at identical twins, studied for decades. So think about it for a minute. If you have identical twins, if you are an identical twin, what is the likelihood that you're going to look very much like your twin? Very high, right? Now suppose your identical twin gets hit by a truck. What's the likelihood you're going to get hit by a truck? Probably pretty low, right? Because that's environment. Now, environment is a tricky thing. It's not just the truck, it's bad luck, plus the truck, right? And that
It's bad luck.
that you got hit by it.
You guys should just stay away from it.
And we call that stochastics or something like that formally, but it's basically bad luck. Now, apply the same sort of reasoning to cancer. Pancreatic cancer, for example. If your identical twin has pancreatic cancer, what is the chance that you're going to get pancreatic cancer? Think about it for a second before I tell you the answer. It's 3%. How about ovarian cancer? 3%. Almost all cancers are somewhere between 2 and 10%, which is higher than if your identical twin does not have cancer, but still certainly not deterministic. And it's the same with other diseases, although a bit higher. With cardiovascular disease, stroke, diabetes, there's certainly a hereditary component, but it's a minor component compared to environment plus bad luck. Now, that may be bad news for geneticists like us because it means our powers are limited. However, it's very good news for the population because think how frustrating it would be is if your fate was written into your genetic code. You would have nothing to do to make yourself healthier or less healthy. And because so much of what happens to us with respect to disease is environmental and bad luck or good luck. It gives us a chance, a real chance, to intervene in the process. Now, in answer to your question, probably one of the great challenges now is to understand how genes and environment go together. We won't understand this bad luck. That is beyond, right? That's just throwing the dice. But certainly there's much to be learned about the environmental component and the genetic component.
If I can follow up, it's an interesting point that people measured the heritability of death due to cancer and death due to infectious disease, premature death in both cases. And very surprisingly, a fundamentally genetic problem like cancer turns out to be less heritable than a problem where you would think luck is involved so much, like whether someone coughs on you, let's say.
Guys, we have a fundamental divergence as to what we mean by the reason for studying genetics. Some people think when we talk about the genetic basis of disease, our interest is in predicting who is going to get the disease. That genetics is here to assign risk. I'm not terribly interested in that. Genetics is here to understand mechanism. Genes and environment work inextricably together. And trying to distinguish one versus the other isn't very helpful. The reason why there's so much focus on genes right now rather than environment is we have wrestled genes into submission. It's a finite list. We have a whole genome. We don't have a comparable way to make systematic, comprehensive studies of the environmental factors, and therefore we're studying the genetic factors. We can't think that because we're studying the genetic factors, the environmental factors matter any less. And it may even be that in a disease where environment plays a huge role, studying the genes will get us to the mechanism, and knowing the mechanism will help us understand the environment. So I really think the public is often confused when we talk about genetics as deterministic or enlightening as to mechanism. But look at what's happening environmentally right now. We have a huge explosion of diabetes and heart disease in Asia. India, China are experiencing skyrocketing rates of diabetes and heart disease, and this is no change in their genes. This is a result of changes in diet, adopting Western diet. Look at cancer. We can debate all we want the genes for lung cancer, but a really great way to get lung cancer is to smoke. And yet lots of people still smoke, and we know that. And we're not so good taking this clear piece of environmental knowledge and translating it through some translational science in getting people to stop smoking. And yet there are other ones where we don't quite know. Multiple sclerosis. If you're born in a northern latitude and grow up in the first 15 years of your life, you have a much higher risk of multiple sclerosis. Why? We don't know. The genes may help us understand that. So the genes versus environment, I don't buy. It's which in any given problem is going to give us the best purchase on understanding the problem and then on doing something for public health.
And how are we going to figure it out?
Well,
the interactions, the multiple sclerosis between your HLA and the cold climate.
Look, all scientific problems involve some combination of hard work and luck. And right now, if we can use the genetics to drive it down to the end and understand all of the genetic factors, we could then ask, okay, if these are the HLA types, what might they interact with? And some smart person is going to come up with an efficient way to take zillions of viruses in the environment and interact or look at their signatures. We've got to count on a next generation that's going to keep inventing tools to use our genetic characterizations and cross it with the environment. And I don't know what those will be, but unless that's done pretty efficiently, we're never going to make the same rapid progress in environment. And we're going to need it with regard to physiological mechanisms, because we trigger these. Autoimmune diseases all over the world. Type 1 diabetes has increased secularly over the course of the 20th century. So there's a lot that's going on, and we need to understand those mechanisms.
So, well, while we're waiting for the next Eric Landa to arrive on the scene to figure out the environmental aspect, which I agree with everything you say, we do have to deal with patients that do have these diseases that have strong familial components, like in Bert's area, colon cancer, as you point out, the chance, if one twin has colon cancer, the chance of the identical twin having it is going to be probably less than 10 to 15% over the whole lifetime. So a colonoscopy is much better than having your genome sequenced. And I think that point has not sort of reached the public to try to understand about colonoscopy. It certainly hasn't reached these companies that have, like 23andMe and other companies like that advertise in the United States about how one will learn about genetic risk. So I think that's an important aspect. And in heart disease, which I know a little bit about, there are, like 42 different GWAS or SNPs that have been identified in large studies that predispose to coronary disease. Each one has a very small effect. But when it's all said and done, the best prediction to have a heart attack would be a family history, an LDL cholesterol level, don't smoke, and have your blood pressure measured, and take a baby aspirin. So in that, I think those 5 things have led to, over the last 60 years, about a 60% decrease in deaths from heart disease in the U.S. and also, I think, in Europe, too. At the same time, as Eric mentioned, while this incredible drop in deaths from coronary disease is coming because of prevention, which doesn't require more than 2 or 3 things, there's this unbelievably increasing rise in obesity and type 2 diabetes. And what people like Richard Pito and very smart epidemiologists predict is that in the next 5 to 10 years, the downward slope in deaths from coronary disease is going to change, and it's going to start moving up unless we do something about the obesity and the type 2 diabetes. So we have a real problem in terms of the changing dynamics of chronic disease. And cancer is also turning out to be a major consequence of obesity. In fact, some people think, Dr. Varmus, the head of the Cancer Institute, says that he thinks that obesity, arguably in the next few years, may be as important as smoking for cancer. So these are sort of important things in thinking about prevention when we don't know everything about what the precise environmental factors are.
I sense an interesting tension here between Joe's more classical approach and Eric's genomics approach to medicine.
No, not at all. I mean, I completely agree. We know a bunch of things. We need to act on those things now. And it's stunning how poor we are at acting on the things we know. At the same time, I don't think Joe and I disagree that we need to discover a lot more of them. For many of these, where we know there are environmental factors, we don't know the factors. But for some, diet and exercise, boy, that would make huge differences to the rate of obesity and type 2 diabetes. We know this fact.
So is it diet and exercise that's missing in India, where the incidence is skyrocketing?
There's a changing diet. There's an extraordinarily changing diet with prosperity. And it is going to have an epidemic proportion unless there is a change in behavior to go with it somehow. And, one approach would be create drugs to go fix all this. And another approach would be to actually get the problem at its root, as Joe says. So I don't disagree at all.
So perhaps I could ask a question to crystallize this, because the topic here is genes and society, not just genes and scientists. So, as Joe said, for colon cancer, the identical twin rate is 7%. It's less than 10%. So if you were given $1 billion to make an impact on colorectal cancer, knowing that 7% is heredity and 93% is environment and bad luck, roughly, right? How would you spend that? Would you spend it on more genomic studies, on more epidemiologic studies, or more biochemical studies? And I would ask you the same question, because...
So Bert, you got to tell me if I'm investing.
Yeah.
Then economically, I need to know what's called your discount rate. That is, I need to understand how you discount the future. If, for example, your discount rate is very high and you wish to have return in the next several years, I would invest the portfolio one way. If we were dealing with polio, I'd invest in a lot of iron lungs, if we were in the 1950s. Right? On the other hand, if your discount rate was a social discount rate that said you care about these years plus the next generation, I might be thinking about vaccines for polio and things like that. So
that's what I'm asking.
So do I get not only the $1 billion, but get to decide what the social priorities are across time?
You get to decide everything anyway.
I would like to... Before the panel is over, I'd like to have that in writing. Thank you, Bert. I appreciate it.
But let's talk about the long-term use.
But let's name a problem. Pick one, because it'll depend on the
colon cancer.
Colon cancer.
Yes, I want to wipe out colon cancer in the next 50 years.
Okay, so that's an easy one.
Okay.
I would turn to a world expert on colon cancer and say, Bert, as the world expert on colon cancer, what would you do?
Joe?
Well,
you're going to turn to the heart disease expert?
No, I just want to see his answer.
He's already solved heart disease, so let's give him a chance at cancer.
He solved that one, right?
Yeah, well, colon cancer. So what? Okay, what about the microbiome, which we have not discussed? Maybe that's the key to the environmental aspects of colon cancer, because there's a lot of information with deep sequencing that shows you actually the importance of technology. That's why we have to have good technology, because we wouldn't know about the microbiome. People are beginning to learn that there are different organisms in our intestines that predispose us to different diseases or even protect us. And I think the paradigmatic one would be Helicobacter and peptic ulcer disease. And that was the one for which you all awarded a Nobel Prize many years ago. And there hasn't been one quite that clear-cut, but now there's a huge amount of information. There are proposals that there are certain organisms in our nasal sinuses that protect people from sinusitis, whereas people who have chronic sinusitis don't have these organisms. And perhaps there may be a chance of introducing those organisms. And so coming back to colon cancer, maybe that would be a key to find out whether people in the early stages of colon cancer have different microbiome in their colons.
Okay, so Joe is doing the genomic approach, but to the colon cancer expert, suppose there was a non-invasive monthly colonoscopy. How much difference would it make?
All cancers, including colon, the deaths would plummet by 90% if you could detect them early.
So maybe Steve Chu from The Morning should have some kind of an imaging thing, and we ought to be able to figure out, if you really think that that would be the most effective thing, then everybody takes a pill that takes pictures along the way, reports it out on the internet, and every month your colon is checked.
That's one of many options. I mean, my answer to the question I asked you would be a combination of understanding colon cancer, both at the physiological microbiome level, but because I'm interested in the disease, not just as a very interesting topic to study, but because people die from it, I would say at this point, any projects that strive for prevention, since it is totally preventable or almost totally preventable, should receive a lot of funding and research. And I believe, in answer to Garen's question, I mean, part of the reason it takes so long to get from here to there is because as a society, we react to these crises. We try to cure these advanced cancers. When we talk about curing cancer, we're talking about curing advanced cancers. We're not talking about preventing cancers. It would be talking about iron lungs and polio instead of vaccines. So it's not just scientists, it's the way society is thinking about it.
Very clear. One of your breakthroughs was really on the BRCA1 gene that's led to prevention to some extent against one type of cancer. Sure. How do you respond to the question that Bert asked here?
Bert is really the parent of this field, and what we did in breast cancer was to take the paradigm that Bert had established for colon and apply it to breast, which was to say, if you ask a question about heritability for a population as a whole, you'll get an answer between 3 and 10% for any cancer, any common cancer. Retinoblastoma would be higher, but for the common cancers, it is exactly as Bert said. But Bert had established the paradigm that if you work particularly with families that are severely affected, you may be able to isolate individual genes that have a major impact on people who carry mutations in them. And of course, that proved to be true for BRCA1 and BRCA2, and now 20 or so sister genes. And we can and do prevent the breast and ovarian cancers in those women, but it's draconian. I mean, this means that a young woman, when she's finished having her family at the age of 35, 38 or so, undergoes a prophylactic removal of the ovaries and fallopian tubes, which of course reduces her ovarian cancer risk and also reduces her breast cancer risk by about half. Virtually all these young women do take some replacement estrogens, which are titrated carefully by themselves and a good gynecologist for quality of life until she goes through a natural menopause. So we can and do prevent, for those women for whom we know the inherited mutations, we can prevent breast and ovarian cancer. It's a source of grief to me that we can't yet make the technology that would make that mutation screening available to all women in the US. You're doing a much better job of it here in Europe. But there are both logistical and economic reasons that preclude it. But I'd like to step back a moment from all of our discussion. We're all geneticists, and so I'm going to take the non-geneticist view here for just a moment, just wear a different hat, just the citizen view, which, of course, is the other role we all play when we're not actually having fun in the lab. And that is that I suspect we would all agree that there is an edge to the way that we think about genetics, which when out in the world can have a real negative impact. And that is that the more that we discuss the role that genes play in illness, the more that there's a tendency to generalize beyond illness to life. When in fact, we as geneticists work around the margins. I think what we try to do is either prevent or resolve deleterious things that happen to people in consequence of their genes. If we don't convey at the same time that most of daily life has got nothing to do with this, that the success of children in life is due to hard work, is due to enjoying what they do, is due to their choosing to a way of life that appeals to them, that is free will, hard work, fun, intensity, all of the things that are in fact a product of something very different than what we're focused on, namely a good public educational system. If we aren't pushing that agenda in parallel with simply stating the reality of what we do every day, I think we're missing a beat. It's a straightforward message for all of us. We all know it. All of us grew up in good public education systems. Every damn one of us, right? We're all middle-class kids. As Jim Watson says, this field is the middle class. We are kids of parents who had the money to send us to college, but weren't so rich that they felt entitled. I mean, we are the kids of Depression-era parents. And if we can't convey to our constituencies, whatever country, that if we lose that one generation social mobility that is the consequence of fine public education, we lose all of this. And that working around the edges with the most advanced genomic technologies won't gain us a thing if we don't treasure the ability of the child to be able to choose a life that they can work hard at and love.
I think we probably all agree about that. Okay.
We do. I'm not criticizing. We all do. Yeah.
Yeah. Joe?
So one thing back, just thought about it to Bert's question about what would you do? What about cancer, circulating cancer stem cells? Is this something for the future where one will be able to go into the blood and identify a single cell that has a RAS mutation, a P53 or AKT? Will this happen?
Yeah, and I think Eric mentioned that one of the beneficiaries of genomics was somatic cell genetics, which are mutations that occur after birth, so they're not hereditary, but they play the predominant role in cancer. That's what distinguishes cancer from all other diseases. And there are efforts ongoing now, as Joe mentioned, to look for either cells or DNA in the blood, again, as a way to detect cancers early. I mean, for colon cancer, as an example, there are 55,000 deaths in the U.S. this year. All of those people will die only because their cancers weren't detected in the first 90% of the cancer's existence. Cancers take about 30 years to grow, and all of the people that die from metastatic disease. Metastases only occur in the last 2 or 3 years of the cancer's lifetime. So technologies like that, so based on sophisticated DNA technologies, cell sorting technologies, have an enormous potential. Proteomics made huge advances, but still have yet to be applied, and more importantly, have yet to garner the resources from NIH and other funding bodies, because we as a society are so focused on curing these advanced cancers that we often don't think that there's other ways to tame the beast.
It does seem like one could have a panel of mutant genes and abnormal proteins that would screen for the most common tumors that would be applied every year in a routine test. And I know that would seem to me as important as anything else the NIH could do right now.
Well, but the NIH has got to be prepared.
You look for necrotic DNA from necrotic cells in blood.
I mean, remember,
There are any number of ways you could look for markers, but let me throw out what we have to be prepared for, which will be, so I very much endorse this, but what you'll probably do is you will catch 500% of the cases.
Right.
Yeah.
Because you will find that there are a lot of instances where there are these suspicious cells or some DNA running around in a mutant state that is already going to be dealt with by the body. And so we need to be able to be prepared to say that to the public, that we're going to, at the beginning, we're going to over-alert, and then we're going to need to figure out why we're over-alerting. It's not a reason not to do it, but we have to be prepared that we swing one way or the other. And eventually the goal is we're going to be able to say which ones you really have to be worried about.
That's where you have to have clinical trial.
You've got to be set up to do the clinical trials. Ideally, you should be setting up now even in advance of the markers so that you could do a retrospective, prospective clinical trial.
Okay, so let me just wrap up this. We are probably in for a very interesting new era of diagnostics, early diagnostics. And the challenges we're going to face are those classical ones of sensitivity and specificity. Do we want to sort in such a way that we catch all real cases or real threats, but a lot of noise as well? Then we need a second test, just like it was set up in HIV diagnostics a number of years ago. If we move now a bit from cancer to other complex diseases with gene-environment interactions, we've seen in genome-wide association studies for instance, heart diseases, metabolic disorders, that they've caught a number of genes, but each contributes a rather small incremental risk, an odds ratio 1.2, 1.1, something like that, and a long number of genes. What does this reflect? Eric proposed, but he's not completely uncontroversial here, that it's polygenicity. Many genes interact to cause the disease. Whereas Mary Claire, in her lunch talk, suggested that we're dealing with heterogeneity, in which case we would need to phenotype these diseases better, that we lump together as heart disease or metabolic syndrome or something like that. So, which one of them is right?
No. But we have the same confusion I raised before comes up again in your question. Are we asking about making predictions for people or understanding mechanisms? Let me give you a gene that has a pretty tiny effect from the point of view of prediction. It's a SNP that has a very small effect on prediction, highly statistically significant, but maybe 5–10% increased risk. It's HMG-CoA reductase. Now, you wouldn't be making predictions based on HMG-CoA reductase, but Joe's work tells us that drugs that work against HMG-CoA reductase are enormously powerful. We always confuse ourselves between whether we are talking about predicting things about patients or understanding biochemical and cellular mechanisms.
But isn't the problem, Eric, that if we went for the unbiased approach completely, totally unbiased approach, we would miss both the LDL receptor and HMG-CoA reductase?
Why? They come up.
Yeah, but with a very small number.
No.
Let's hear what Joe says about this.
Well, I think they definitely come up, and there are 95 genes and lipids that come up, and about a third of them are genes that we know a lot about from Mendelian disorders and a lot of research. The issue is whether, if you didn't know anything about any of this, could you come up with a story from these 95 genes? I would say no.
And that's exactly what we talked about at the beginning, is that biochemistry is brought to its knees, and cell biology is brought to its knees by a measly 95 genes. It will look, from a decade from now, silly that we were agonizing over, oh my God, we found 95 genes in a process and we couldn't figure out what they're doing. It's a statement about our inability right now to make sense of those things. When I quote schizophrenia, and I look at the 62 genes that were reported at the recent International Congress on Schizophrenia Genetics, 62 hits, 4 are the subunits of the same L-type calcium channel. That's no accident. It tells you.
That would be a good. That one would be a good.
That one might be a good one. But it's our inability to read this stuff. I don't disagree with you that it's hard to make sense of it right now, but we shouldn't tolerate that we don't understand how to interpret this in terms of circuitry. It's a failure of cell biology.
But there is one, like the first polymorphism I learned in medical school, I'm sure Bert did too, and Bruce and Goran, ABO blood group. That was done many years ago from immunogenetics before GWAS, and it's about one allele gives a 1.8-fold increased risk for peptic ulcer. Now, had people used that as a clue to pathogenesis of peptic ulcer, people did try to work on that. They never could get anywhere. You would never discover Helicobacter. And to this day, we don't really know why the ABO blood group actually confers a population risk to peptic ulcer. And that's one that there have been thousands of studies and everybody agrees on it. So there are some red herrings in the way that will deter any type of biochemical study you would do based on risk factors.
Nils, you want to comment here?
Yeah, so I would like to make a little bit of a provocative comment here, as I'm working at an aging institute. So maybe we spend too much effort on working on specific diseases. I mean, all of the big killer diseases, atherosclerosis, diabetes, dementia, cancer, are age-associated. Evolutionary theory predicts that evolution has selected to maintain our bodies in a very good shape until we have passed on our genes to the next generation. We are sitting here now in a protected environment, most of us aging, because there has simply been no evolutionary pressure to maintain our bodies in good shape at this high age. And certainly in experimental animals, you can make simple interventions like, for instance, caloric restriction that prolongs the healthy lifespan and actually counteracts multiple types of pathology. So should we do a bit more basic aging research and a bit less of the... It would be interesting to hear what you think about these thoughts.
Who would want to take a shot at that one? You could argue, though, that it's been rather successful to go for specific diseases. It's pushed up the average age from 35 to 85.
Yeah, but I mean, there are also demographers predicting, at least some of them, that if you would cure all cancer, all atherosclerosis, it would only prolong, of course it will decrease a lot of suffering, but it would only prolong human life with few years, right? So if you solve a problem...
Few years times 7 billion people is not so bad.
No, but it's not bad. I'm not saying it's bad, but I'm saying that multiple things happen in aging, and if you cure one problem, another one tends to appear.
My
right?
goal for a BRCA1 carrying young woman is that she get to the point that matters. If we can prevent premenopausal breast cancer, I think we've done it. You know, that will be for me a success story. The question of cellular aging and building enough biology behind our understanding of aging is, I think, the next great challenge that dwarfs all of the other challenges we've been discussing. May I go back to your previous question about the debate between polygenicity and heterogeneity that I mentioned at lunchtime? For mathematical reasons that I won't go into on a beautiful December afternoon in Stockholm, I reject a great deal of the results of GWAS. I believe that this was an experiment that needed to be done. It was a good idea. It was a good theory. I think for the great majority of applications, it has proven incorrect for mathematical reasons, not for reasons of stupidity. They were brilliantly done experiments, but I think that they have shown us very, very little. I think it won't matter. I think it already doesn't matter, because with genomics becoming as efficient and effective as it has, there's no longer any need to pool phenotypes at all. We can look at the individual, and we can look at the family, that is the small nuclear family or the kindred. That it is essentially now moot whether we must do an analysis across an entire population or whether we can only look at Mendelian disease. We can have it all. It's almost as if Eric had put into his talk this morning a third way, right? There is the family with Mendelian disease. You said this morning, there's the family with Mendelian disease. There's the population with complex disease. I would say, baloney. We are beyond that. We can now look at the individual in the society using genomics and understand if this person does or does not carry serious major alleles for any of the diseases.
Let me agree and disagree.
Let's hear what Bruce says about this. You've done a lot of focused genetics.
Yes, well, we've always concentrated on monogenic phenotypes. Yeah. We've had a lot of success with it.
You've done pretty damn well.
And I must agree with you, overall, I haven't seen the payoff from GWAS as an approach. Right. And I think most everyone would agree with that.
Well, maybe. Not in my world.
Now let's give Eric a chance, and then we have a question through the web here.
Look, I think we're talking about a rather false distinction. We're talking about the frequency of alleles in the population. What you call GWAS is looking at higher frequency alleles, medium frequency. Yes, you. Yes, you. Because remember, genome-wide association, you're going to do an association for rare variants, medium-sized variants, and common variants. It's
you may redefine it however you like, my dear.
No. Mary Claire.
I know what you've been doing.
Mary Claire. You are, in order to find that any gene plays a role, unless you have a single Mendelian family in which you can get strong transmission, you're going to need to see, as you do in your studies, multiple hits in that gene.
No, not true.
Well,
I mean, granted, I
would
love to see
offline, but in any case.
See it.
Sorry?
I think we'll perhaps stop there and agree that we disagree, and that maybe GWAS is not the final word on disease.
Multiple hits in a gene. And you're using
okay, thanks. Now we have a question from...
Pardon me?
Do we have a microphone? Yeah, you can fight it out later. We have a question or two that came through the web here. Indira.
Help me out.
So this is a general question that's shared by the online audience from Mumbai to Massachusetts. It's, is it possible to silence an extra chromosome at an embryonic level or even simply repress the expression of a gene to prevent the manifestation of diseases and syndromes?
Great question.
Who would like?
Bruce.
The question seems to be referring to germline engineering, and in principle, is it possible to do? Yes. Is it desirable to do? That's something debatable, but in principle, yes, it should be possible.
Mary Claire, I mean, it's related to PGD, so you might want to say something about that.
That's a very good thought. I hadn't thought of it in that context. What a good question. Yes, this was something that I mentioned at noontime, that there is now the possibility of wedding genomics to pregestational diagnosis. And that is that we can, for an individual family, and in this case it is very much a family-based analysis, we can determine what the particular mutation is, wherever it is in the genome, that has caused an illness in the family that a young woman, of course, wishes to avoid in her own next pregnancy. Then when that mutation is identified based on the people in the family who carry the illness, we can determine if that young woman does or does not carry the same mutation. And then the gynecologist can use pregestational diagnosis. That is, they can take eggs from this young woman, they can take sperm from her husband, they can make zygotes in a petri dish, and they can genotype off of the Barr bodies or off of the extra cells the particular genotype of every zygote and of every 16-cell embryo, and then implant back in the mother a healthy embryo. So a healthy child is born. There's no abortion involved. This is within the constraints of every religious tradition, and it is actively being done now for conditions for which the genetics can be determined and for which there is an established link already between gene and phenotype, which is a very large number of them. So in a sense, that is a response. Thank you, Bert. It is a response to the question of your correspondent.
Do you have more questions? Yeah, please. We have a question from the floor.
Okay, wait.
Could you microphone?
I think you explained the selection for healthy zygotes. I think the question there was that if you can correct the problem, if all of the zygotes has the mistake or mutation.
Right. Okay. So it's a far more difficult question. Well, I pass it to you because it's harder.
Joe gets the difficult questions.
I think that, I think that what Bruce said, in theory, you probably can do it, whether or not it will be desirable, but you could, in theory, silence a mutant allele if you knew, for example, in Huntington's disease, a certain allele carried the tendency to have an expanded repeat, then you could, in theory, be able to silence that prenatally. That's a long ways off, but it could, and there are other examples too where silencing deleterious alleles, especially in dominant conditions.
But we have no idea how to do it.
What's that?
But we have no idea how to do this.
No, exactly, right.
That's important to say.
Great challenge for the future.
It's a next step challenge.
But it is impressive that one, getting back to circulating cells, can diagnose chromosomal disorders.
Yes.
And prenatal diagnosis on the basis of a single cell in the mother's blood. And that technology is very advanced now.
We have a question right there.
Does it work? Yeah. I don't know if any of you want to comment on the fact that Europe very, very recently approved the first gene therapy from a company in Netherlands, if I don't say wrong, which looked like a boom. I read it through the webs, and it was also features in Nature and so on. And then you go a little bit in detail, and I was like, yeah, it was approved, but it was a lot of concern. And yes, only for this very tiny group, and it was for this very, very tiny group, in that very tiny group of diseases, which will eventually target like 100, 300 people. I wonder...
Could you condense the question?
Sure. Do you think, it's related to what came out from the web, gene therapy is a great promise, but it's quite far away. How far away are we from safe gene therapy?
Okay, how far away are we from gene therapy as an important clinical reality? Bert, sorry, you got it.
I would be surprised if by the time that our children were our age, that gene therapy for certain diseases wasn't being instituted. I would suspect the first one they'll be instituted for, other than these rare diseases that you mentioned, are diseases of the eye. Retinitis pigmentosa, for example, in which it's possible to perhaps correct a few cells or a small fraction of cells and recover a good amount of function. For other diseases, such as cancer, it will be extremely difficult because there you have to essentially correct every cancer cell. But for diseases like retinitis pigmentosa, for certain blood disorders, in which you can do the gene therapy ex vivo and then infuse them back, I would be surprised, given what's gone on so far, that that doesn't become routine.
Gene therapy has been a rocky road in a way in its development, but for the lay people in the audience, we shouldn't forget that DNA technology has provided many new drugs, recombinant antibodies, recombinant proteins that are used every day in every hospital around the world. So it's been a revolution. The next challenge is to correct genes, and it turned out to be a bit more difficult than we had expected based on problems with delivery and of transformation of the cells. Now, yeah, Deepak Srivastava.
Thank you. Earlier in the discussion, Dr. Goldstein mentioned the importance of better human models, say, compared to mouse models. And Dr. Lander commented on the need for better high throughput or new technologies for functional discovery. I wondered how this year's Nobel Prize selection of iPS cells will impact those two areas.
Well, I think it's a good question. I think that as one gets better and better making iPS cells differentiate into the cell type that you want, especially in patients with different diseases, they become very good models for high throughput screening for small molecules that are able to correct the defect in the cell.
Yes, but. So I want to agree with Joe, but again, I want to exercise both caution and optimism here. When you take a patient and you make iPS cells, you're getting their whole genome with all the variation there. I think when you do that, you don't exactly know what that combination in the genotype is doing. What encourages me is combining that technology with some of the genome editing technologies that have come along right in the past 2 years even. The ability to introduce in a very effective way selective edits so you can make isogenic iPS cells and know that you're studying a real difference. Then I think when you combine those two, we can get at whether we're hitting that mechanism or inadvertently one of the hundreds of other differences that may be present in the patient. So I agree, but as with all these things, as with gene therapy, it took a while to get it really rigorous and it'll still take longer. I think iPS cells will be fantastic for that reprogramming, but it will need a whole battery of technology around it to get really accurate answers.
And also, this determining what phenotype in an iPS cell, for example, in chronic neurodegenerative diseases, that's where these are going to be used, like ALS, amyotrophic lateral sclerosis. What is the right phenotype to produce in an iPS cell that will be most reminiscent of what happens in vivo? Those are going to be the real challenges.
So we've covered quite a lot of ground, and we have just a few minutes left. We've discussed genomics quite a lot. Now I have a question to the audience. How many of you would like to have your genome sequenced? And then we'll check with the panel what they think. Hands up. How large percentage is this? Eric, you're a mathematician.
We're all failed mathematicians.
30%.
This is about
2/3?
60%.
Yeah, he's an optimist. Joe says 30 to 40%. So I think 55.
This may have to do with vision. Hold your hands up again. Let's see. Okay, so this is yes. Now put them down and do no.
No, it's better. It looks about 50. I think
50-50. Joe and I are compromising at 50-50.
They finally agreed about something. That's wonderful. Now, Mary Claire, would you like to have your genome sequenced?
I have no strong feelings one way or the other. I haven't done it. I mean, I could sequence it any day, right?
Sure. I know you could.
I haven't because the money is better used. It's $5,000 to sequence a genome now in my lab. I'd rather use the $5,000 to sequence the genome of someone who presents with a phenotype that we need to solve. I have no objection to it at all, but it's not a high priority.
Ms. Göran, are you using Max Planck money to sequence your genome?
I mean, I want like to have my genome sequenced. So what worries me is when I leave the room here, Eric Lander will go to my chair and collect hair, dandruff, and then. I mean, I think it's since you need so little material. What really worries me is also the personal integrity. How are you actually going to ensure that your genome is not sequenced, even if you don't want it to be sequenced? I mean, I think it's impossible to protect oneself. And what will happen? Are insurance companies going to ask for genome sequences? Or are you going to hire a CEO only if you have sequenced the genome of this person? I think there must be some legislation, some thoughts about this.
Joe, how do you hire a dean in Dallas nowadays?
Well, I don't think I would learn that much from my genome sequencing. I was sure I could learn whether I had red eyes, because there's a single gene for that, but I don't have.
It might be.
I mean, red hair, not red eyes. So I would say, if one got an illness that was difficult to explain, not necessarily cancer, because that would be a somatic, you might decide to do it. But I would say right now I would not.
Bruce?
I'd probably be inclined to say yes, why not? But on the other hand, I understand all the news that you ever get is bad.
So you have not done it?
I haven't done it.
Eric, you've done it already, have you?
No, not at all. No. I haven't sequenced my genome, had anybody else sequence my genome, and I have no great desire to. I think, in fact, we send the wrong message as scientists engaging in personal genomics about ourselves. It is about trying to understand. It's not about ourselves. Oh, it's technology.
This is just to show that it's not about ourselves.
Yes.
We're sorry.
It is about learning the mechanisms of disease. So we will sequence thousands of people with a particular disease to learn about it, but doing it for one's own individual benefit, right now there isn't that much you would act on. Now, if you said, God forbid, I got cancer, would I sequence the tumor and the normal? You bet. Because I think there are actionable things I would do. So I draw that line there, that right now my germline genome, not that interesting, sends the wrong message. Cancer, I would do it because in some non-trivial fraction of cases, it's actionable. What do you do, Bert?
Bruce.
Bert.
Sorry.
Well, I've sequenced half my genome, put it that way, because I agree, I think, with most that sequencing mine I wouldn't be interested in. But when my brother had a child who had a childhood cancer of a type that's known to be inherited as a single gene monogenic disease, I sequenced the child's genome as well as my brother's and his wife's. Because for obvious reasons to see. Right. But, in special cases, it's clearly important and can be useful.
So it sounds like we've reached a consensus here that sequence when there is a medical reason for it.
Right. Exactly.
And
I think we're being chased.
With that, I think we'll stop here. The panel has agreed to agree and to some extent disagree, and that's stimulating for future discussions. And now over to the next session of this stream, which will deal with how to reach the promised land that we've touched upon during this first part. So thank you very much. The floor to the panel.
Sorry?
Good afternoon, everybody, again. Hello. So, welcome back, although I think most of you stayed where you were. Welcome to the second half of Stream 1, which is called, as you see, The Promised Land of Genomic Medicine, How Do We Actually Get There? And in discussing what we're going to do on this panel with some of the panelists, a couple of alternative titles were produced, if you like, for the session. If I quote from some of the notes I received. And Janet Woodcock, who is here, who is the director of the Office of the Center for Drug Evaluation and Research at the Food and Drug Administration in the United States, said that what she was most interested in is how to translate research findings into clinical practice. And I think that makes a beautiful subtitle for this panel. Juan Enriquez, who has also joined the panel, who was the founder of the Life Science Project at Harvard Business School and is now with a venture capital organization, put it more starkly. He said he wanted to address the disconnect between what we can discover and what we can bring to market. And I should mention that our other new panelist is Tikki, who is a professor at the University of Singapore and was formerly with the World Health Organization, as I have to look here, as director of the Research Policy and Cooperation Unit. Those subtitles, I think, perfectly encapsulate what we're trying to do here, which is to ask how we get all these research findings to make a difference to people's lives. But we did call the session The Promised Land of Genomic Medicine. And I'd like to start by asking our panelists to define what they mean by the Promised Land, or what they think of when they think of the Promised Land. So perhaps we could start with you, Joe. How do you see the Promised Land of Genomic Medicine?
Well, so I thought about this because you had it in your title. The Promised Land of, let's say, personalized medicine. I'll say personalized medicine would be that somehow if one could convert genomics in such a way that every physician and every illness could be treated as it is when we go to the optometrist to have our eyeglasses checked. Everybody in this room has a personalized prescription for their eyeglasses, and I think that's the epitome of personalized medicine, and I think there's nothing actually quite like that, and that's what the aim ought to be. Whether or not that'll be achieved in the lifetimes of we panelists, I don't know, but I ultimately, being a positive person, would say it will definitely be achieved at some point in time. So that would be the idea, that you would really like to have medicine based on your genetics and your, not only on your genetics, but also on information that's conveyed between you and your physician, personalized in such a way that you're treated and have your health restored as well as possible.
That would require a shift in thinking such that people would be happy to have their genome sequenced, unlike all of the previous panelists.
That is true, but you may not have to have your genome sequenced, because it could well be that one will have biomarkers in the blood and things like that. Mm-hmm. In other words, much like the optometrist uses a chart and a few little manipulations with his hand. So,
Janet, how do you see the Promised Land?
Well, as a physician, I can tell you right now, one of the biggest barriers to treating people is there's such huge variability in response of human beings to any given treatment. That's why we have to do clinical trials, okay? Because only a few fraction of people usually respond, and some of them get terrible side effects, all right? We don't understand the basis of human variation, either, as the previous panel said, in disease, but then in response to treatment. So that's one thing, is starting with genetics, but we're going to have to build upward from there, understanding human variability in disease and treatment of disease. The second, though, is we were talking about this earlier, because I think in some ways that one of the biggest promises genomics is not in a human realm. It's to understand microbial organisms, particularly those that colonize us, or actually those that infect us. And probably for drug development in certain areas and for the developing world, I think rapid diagnostics that are point of care is a promise of genomics, I think, that the technology itself. And that could transform the way we interact with, say, infectious diseases, and develop new interventions for infectious diseases. And that would be just tremendous.
Thank you. Tikki, how about you?
Well, after 13 years working for the World Health Organization, my promised land is the 3/4 of the world's population who live in low and middle income countries, the developing world, and the 30% of those people who live on less than $2 a day. And I think the objective for genomics and the genomics revolution is to what Helga referred to earlier. How do you avoid the genetic divide? I think there's a huge inequity, social justice issue here.
Indeed. And I hope that's something we will be dealing with a little bit on the panel. Eric, do you want to have a go?
Yeah, I have much more modest ambitions. I don't like this personalized genomics because it suggests, like with the optometrist, that you're going to go in and get something ground for your own personal genome. That works really well if the cost of personalization is pretty cheap, as it is for the eyeglasses. But getting through truly personalized therapies through the FDA, and it's not the FDA that's the issue, it's getting enough investment behind something highly personalized, strikes me as not so likely. What I want out of this genomic promised land is that we give each disease its right name and we understand its mechanism. If we did nothing but that, but we split diabetes into type 1 and type 2 diabetes, and we split lung cancer into 7 pathways, and we knew which ones were operable, and then we pulled off our shelf drugs that worked against those pathways. Medicine has always been splitting and lumping and splitting and lumping. If genomics gets us to the end of that, by all of the tricks that we've got, the tricks of sequencing common variants and rare variants and microbiomes and combining big data across tons of patients, and we give each disease its right name and its right mechanism, then it will become the responsibility of medicine, the private industry, and regulators to be able to produce the best pharmacopoeia to match that. That, I think, is achievable, and I fear that when we tell the public we're going to give them personalized medicine, we may be setting an expectation beyond what we could deliver on.
So far we have individualized medicine, we have the majority of the world's population being well served, and we have a redefinition of disease taxonomy.
That's all, but maybe there are 2 other things it could mean too on the panel.
Bert, you want to chip in?
Well, I don't think I can improve on Joe's vision, but I would couple that with accountability in the sense that now we give drugs, and we don't really measure in individual patients what the concentrations of those drugs are in their circulation. So for a true era of personalized medicine, I think it would take what Joe suggested, coupled with further analyses of patients to see whether what you predict will work. Or what will be achieved is achieved. And I think a lot of gains could be done even now with that because we give the same doses to everybody, basically.
Well, if I may interject,
Yeah.
I mean, that's already started. So FDA has in the labels of many drugs to look at the drug metabolizing enzymes and to adjust doses based on those findings. Some of those are genetic tests. They used to do other kind of tests, phenotypic tests, but a genetic test is much faster, okay, much more clearer results. So that's happening, and also we have recommendations to test certain drugs because the community has figured out what gene confers risk for both liver failure or for serious skin side effects like Stevens-Johnson syndrome. Those are conferred by various alleles that are genetic. Now, not everything's going to be that simple, but at least we have started down that path. of separating out, understanding both benefit as far as dose exposure and understanding the risk of these rare serious side effects based on genetics.
I'm glad that's happening, but I think what I meant was that we followed up with actual measuring drug concentrations in people after the drugs are given, which is done in early phase 1 trials, but not routinely.
It's too hard in clinical practice, but if you can predict exposure based on the genetics, then you have that information, you can adjust the dose.
I actually don't think it's too hard. I think with mass spectroscopy, it can be done. It's expensive, and you're asking about a dream world.
Exactly.
Not today's.
Oh, okay, yeah. Fair enough. I mean, we had that before, therapeutic drug concentrations, and it just didn't work. Clinical medicine did not take that up.
I suppose it's sort of obvious that in order to reach the promised land, one has to redefine healthcare systems totally anyway. Right, so we sort of all accept that as a...
Could I respond to one thing Eric said, which I think is a very good goal, is to split phenotypes, which will help you get the right mechanism. And, you know, this is going to fall on the role of the clinical researchers more than the genomicist, because they've got to be able to split type 2 diabetes into 6 different categories and say, Dr. Lander, here is gestational diabetes, here is type 2 diabetes is thin, here's type 2 diabetes that has a family history of multiple members, and that's what has not, that's not been done.
And this is a real problem, because genomics alone without good phenotype is much less powerful. Phenotype without the genomics less powerful. Unless both sides are really talking, we're not going to get there.
Yes, there's a consortium that is doing what is the genetic basis of serious skin side effects, okay, generally. They had to get the clinicians together and force them to develop some consensus definitions, both symptomatic as well as histologic, of what are serious beyond your skin peels off, okay, which was basically what it was before, so that then they could correlate, they knew what they were dealing with phenotypically. So we have to standardize from both ends, I agree.
Okay, before we move on, I just want to get Juan's thoughts on this.
Oh, I'm having fun watching this. you come into
can't get off that easy.
No, there's no free lunch. Come on.
So look, I want to pick up on these last 2 threads because I think they're really important. So let me just underline them and put a couple of footnotes on what the 2 of you just said. My first dream is I want to understand cancer medication as well as I understand hamburgers. And it turns out that, there's some of your friends who are just hateful because they can eat 5 hamburgers and never gain a pound. And I look at a hamburger and I gain weight. And, that's really funny until you begin to understand that most cancer medications are dosed based on how much you weigh. And they're absorbed at sometimes a 20-fold difference. Not 20%, 20-fold. And so you can take something like a generic drug, Avastin, and dose it correctly and have a 50% increase in average lifespan. So the stuff that Bert is talking about is life and death. And getting that dosage right is really important. Now, let me take and put a footnote on what Eric said, because I think that's also really important. It really worries me when somebody of the stature of Eric says, we can solve this problem scientifically, but there's no way we're going to get it through regulation, and therefore we can have the data to do this, because there's a huge disconnect between what we are learning we can do and what we can actually get through market. And you can actually put numbers on that, right? The average cost of getting a drug to market has gone from the tens of millions to the hundreds of millions to a series of other things. And what that does to each of you is to get to what Janet's talking about, which is a personalized medicine regime, we absolutely have to blow the system up. Because we're going to know how to cure various subtypes and subtypes of subtypes in stuff that comes out of Bert's lab and your lab. But we do not have a regulatory system or a production system in the pharmaceutical industry to be able to do that.
Let me make sure we get this right, because when we say we can't get it through regulation, one might imagine, I think incorrectly, that the fundamental problem is at the regulator. I think there may be problems at the regulator, but the fundamental problem is generating data that is adequate to bring that drug to market. That is the hard part. I'm all in favor of fixing some things at the FDA, and with another job I have, I was involved in some suggestions there. But I think there are questions of economic incentives. I think there's questions of organizations of our clinical trial system. I think there are questions about identification of valid targets. And so I think all along the way, many aspects are broken. And it is true that the problem is getting a drug onto the market, of which I think there are failures at each stage, and we need to understand it. But I agree with you. Getting a scientific discovery to market is the rate-limiting step, and dissecting where the issues along the way are is a very important thing.
I completely agree with you, but there's one big missing component of this, which to my mind is the biggest single missing component in this particular debate, which is we're not measuring the costs of not acting. Right? So we're not measuring if it takes us an extra 3 years, or if it takes us an extra 5 years, or if a medicine doesn't come to market. When you're talking about inequalities, think about it in terms of horses, right? So if you put a horse jump at this level, almost every horse makes it over. If you put it at my height, almost every horse, but a few don't. And if you double my height, almost no horse makes it over. And the same thing happens with a hurdle rate for medicines. If we increase the cost of bringing a medicine to market for a whole series of systemic issues, it's not one issue. What we're doing is we're making it almost impossible for a tuberculosis medicine to come to market, for a malaria medicine to come to market, for a dengue medicine to come to market, because it is not a disease that will give you the same type of margin that a baldness cure would give you. Not that I would care about that.
Right. That's economic, not regulatory. That's the market rewards there.
Okay, Joe, one thing. And then...
Well, that in a way relates to the robustness of the drug. If a drug is robust, then you'll have a big effect. And I can recall 3 drugs that were approved in almost record time. In 1966, I was a house officer at the Massachusetts General Hospital, and allopurinol, which is used to treat severe gout, patients that have these big tophi, huge uric acid levels, was approved, and we started using that in 1966. And it was approved 6 years after the first patient was ever given the drug. It was studied in only 46 patients, but because the uric acid dropped from 20 to 5 and the tophi melted away, it got a rapid approval, and it's still used today, 46 years later. So that's an example. And then Gleevec, we all know the story there, and also the ALK kinase inhibitor. I think those are 3 drugs that I can recall that all got approved rapidly because they had very good efficacy with minimal side effects. So I would say one thing would be, so the challenge might be to get better drugs, not sort of a lot of Me Too drugs are drugs that have marginal effects that require thousands of people to prove their efficacy.
Right. We already have enough issues to take us to end of the day. Let's focus on one. Let's start with disease heterogeneity, increased dramatically by sequencing. And poor Janet is sitting here as the representative of not just the FDA, but all worldwide regulators. You're the only regulator on this panel. So how does disease heterogeneity necessarily means changing the size of patient populations, and in many cases it means decreasing the size of available patient populations for clinical trials. How, as a regulator, do you face that problem of less people to test drugs on?
Well, in drug regulation or getting drugs onto the market, there's a series of trade-offs that are taken. A drug that is indicated for a very large population and for a very minor problem has that very high bar, right? A drug that's indicated for a very small population that has no other therapy and has a life-threatening disease has a very low bar to get over, relatively speaking. And a drug that's very effective for people with a life-threatening disease with no alternative therapy has a very low bar to get over. So those trade-offs are at play in this area of targeted therapy. The drugs generally that we've approved, I think 10 cancer drugs drugs this year already. Most of them are targeted therapy, and the industry is moving in that direction. And in many cases, those drugs in that small population, that subpopulation, are highly effective, okay? And so for that reason, they have a low safety bar because they're for cancer, all right? And they have a kind of low bar for drug development because there isn't anything else out there. However, we're going to have to fix our clinical trial system. In the US, I don't know about Sweden or Europe in general, but only about 5% of adult cancer patients are even offered the opportunity to participate in a clinical trial. And so if we want small subpopulations, how are we going to find them if most of the people who have cancer are just being treated with standard therapy and are not even sought out to enroll? So I think there are multiple iterations or problems to this, but I don't think the regulators... Now, if we had a targeted therapy for... say acne, okay, we would want it still, even if that was a certain subgroup of people, we'd still want it to be pretty safe. And so that increases the cost of drug development, raises the bar, because we have a large number of people with acne, and there are alternative therapies for acne available. So the public would be outraged. We put an unsafe acne drug out there, even if it were we have one on the market already that's highly effective and pretty unsafe, Accutane. So anyway, I'll stop. But that's I was just kind of giving the boundaries here.
I just wanted to ask one follow-up, which is, at the heart of this is risk perception by the public, because you're between a rock and a hard place. You're being pushed from one side to get drugs on faster, and you're being pushed from another side to keep dangerous drugs away from people.
It gets to what we're saying about more effective drug, because the trade-off is between uncertainty, how much uncertainty you're willing to tolerate, versus how much benefit you want to get.
But the point I was going to make is that at the center of this is really the patient and what the patient is willing to accept. And I imagine that a big part of this, and this is to do with, I suppose, the science and society theme of this day, is that that making these hurdles different, changing the clinical trial model, depends wholly on getting people to believe that the change is necessary and getting them enrolled.
Well, I think for the cancer and others, I would point directly to academics in the pharmaceutical industry. Patients want to enroll in clinical trials. He doesn't think so. Well, maybe not at academic medical centers.
I wouldn't argue with that, but I would argue that it's the fault of the pharmaceutical companies or the academics. Now, I don't want to bash the FDA, because I know you have a terribly hard job. You get criticized no matter what. If the drug works, doesn't, you get criticized. It doesn't matter. So that's not what I'm after. But let me give you one example of a clinical trial that we're doing in academia. We have a drug. It doesn't matter what it is. But it works extremely well in experimental animal systems. A week ago Friday, we gave the drug to a neurosurgeon who had metastatic cancer, and he had failed all other therapies. Now, he knew exactly the risks and benefits of this drug. But because of the way the clinical trials are structured, we had to give him a dose 100,000 times lower than the dose that works in any experimental animal. And that's true of, you have to start with a very low dose in clinical trials. And this is partly because the whole process has become institutionalized, and because there indeed have been some egregious errors and unethical behaviors. Just the scale has tipped the other way. We have gone too far. Part of the reason that people don't sign up for clinical trials is they're smart. They know that if they're in the first patient enrolled in a clinical trial, they're going to get a homeopathic dose. They might as well get water, right? Why do we have to start so low? As long as their education, as we educate these people, and let them decide their own risk. They're the ones that have cancer, not us. If they want a higher dose and are aware of the risks, why not give it to them?
Right, tick, problem solved.
So how far does that go, Bert? I mean,
Should we just then make those drugs available without approval at all? Because if they want it, they should be entitled to it?
No, of course not, because
Okay,
all of these drugs go through IRB
so you just want to
approval.
tweak the system by saying higher doses earlier reasonable. No, I'm with you. I'm saying we can make adjustments to it. Good.
Yes.
Can I go to Janet's point, though? Because I think her point about getting the right patients into the right trials, sure, a small number are enrolling in trials, and it would be good to have a larger number enrolling, but the whole concept of enrolling in your trial or your trial is nuts.
Right.
What you should be doing is enrolling in the trial system, getting your cancer sequenced, and then getting into a trial based on the mutations you have. And, you know, it's just there should be a central clearinghouse that says, you're relevant for this drug. Because what we do now is we have a trial, we collect a large number of people, and then if we have a molecularly targeted trial, reject most of them as not appropriate for that trial when they're really appropriate for something else. That's really unacceptable, and we've got to work together. But no one pharmaceutical company can fix that. That is something that requires cooperative action.
I agree, and I think we need, for targeted medicine in general, or genomic-based medicine, we need a big funnel, okay? And then we need to be able to put people into the trial or section of the trial that would be appropriate for whatever their problem is. Cystic fibrosis in the United States, the Cystic Fibrosis Foundation did this. And cystic fibrosis is a very rare disease, but pretty rare. But they had a drug that only worked in 7% of the patients. And yet they were able to locate them rapidly and conduct a trial. A drug is on the market now, and it seems to change the natural history of cystic fibrosis. And the reason they did that is they had all the patients ready, and when a drug comes along that's ready for those patients, they're ready to get it to them, rather than taking a year to go out and get everything together and do a single trial for that drug and so forth. So I think some of the issue, in my mind, I have been thinking for a long time about how to lower the barriers to get to market, but still generate the information that society wants about the drug and that the biomedical community wants before they're willing to use a drug. And I think that one of the ways to do that is set up a system, a scientific system, to evaluate drugs and move that toward clinical practice rather than have them be completely divorced. One's over here, one's over there.
I couldn't agree with you more. Let me paraphrase a former head of the FDA who said, look, I can't count the number of times I've been dragged before Congress to address a problem with an approved drug, and why did you approve it? But the number of times that I've been dragged before Congress to be asked, why didn't you act? Why didn't you bring a medicine to market? Why didn't this medicine come to market? Is exactly zero. And those are the hidden costs in the system, and they're incredibly dangerous, because what we're doing is we're ratcheting these costs to the point where most pharmaceutical companies are no longer being led by researchers, they're being led by lawyers. And most pharmaceutical companies are now spending most of their budgets on either merging with another company or on marketing the bejesus out of what they have, to the point where some single pills have marketing budgets larger than Budweiser, right? Just to put it in order of magnitude. Now, what ends up happening in a system like that is you get less and less drugs to market, unless if you have really innovative technologies like the startups that two of you have started. But if those startups don't survive, if there isn't a financing mechanism, if it takes 12 years, look, this is not an FDA problem. This is a problem that when a institutional review committee at a university says, you know what, let's study this another 6 months. We're not measuring what you were talking about, which is, do 14,000 people die in those 6 months that would have lived if that drug had come to market sooner? And if we start measuring the cost, that's the big missing measure in here, right? How long does it take, and what happens if you don't act? And how much does it cost? And if you don't get one of these medicines to market, it is conceivable that we are killing more people now by not acting than we're saving by regulating. I'm not making that as an affirmation. I'm saying I don't know the answer to that question, and we should measure that.
Thank you. I'd like to shift this discussion slightly to the developing world perspective. Tikki, what are your comments on the design of clinical trials, the testing of medicines, more targeted medicines within poorer nations?
Yeah, I've been very interested to hear Juan's comments in particular about barriers to get to market and the bringing of drugs to market. And I absolutely agree what he pointed out, that there is a market failure, especially with drugs, where pharmaceutical companies don't see a profit. These are diseases affecting people who can't afford to pay for it, basically. And what it means, in my own view, is that the blockbuster model that pharma has adopted has failed in this particular category of drugs. You mentioned TB and dengue as two good examples. So within sort of a global perspective, and the WHO has actually been working with this with WTO as well as with the World Intellectual Property Organization. The challenge is, how do you delink price from the costs of R&D? Mm-hmm. And you mentioned the costs of this. And one idea that's been developing in the last 2-3 years or so is, are there alternatives to finance R&D beyond just protection of intellectual property? And there are some really, really innovative ideas like advanced market commitments, tiered pricing, for example, transfer of technology to partners in middle-income countries, the idea of product development partnerships, patent pools. GSK just put 15,000 of their patents on a publicly accessible patent pool for anybody who wants to use. And a very innovative idea started by the government of France, actually, called Unitate, where every air ticket that's bought in France, the people who buy it are asked to contribute €1, 10 euro if you travel business class. And that's raised something like $600 million just in the first 2 years. Okay, so that's being used now to support or try to overcome such market failures. So there are ideas floating around to get across this limitation that Juan mentioned.
Now, you headed a project to identify grand challenges for the use of genomics in improving public health in the developing world. Was what you just said the finding of that project, or is the finding of that project other than that?
Well, it was certainly one of them in the sense of improving affordability and access. But the other issue that was raised during that grand challenges is something that's been mentioned before, and that is regulatory capacity, especially in the developing world. Now, it would be very nice to reach out to the audience and see if they have some questions they would like to ask the panel. So, are there questions? Yes, please.
Good afternoon. I would like to ask you whether you could comment on the role of the media. Do you think that the media supports your work and the process towards the market, or would you say it slows it down?
So basically this panel is dealing with hurdles that stand between research findings and reaching the patient. Does the media stand in the way or support?
I would say one thing about that. I mean, the media is much more, mass media, not the scientific media, which are very different, but mass media is much more focused on sins of commission than sins of omission, all right? And so the fact that even excessive caution, if you might, by IRB, the whole regulatory system around risk for people who are dying, okay, relates to, it's much better perhaps in the mind of like how you present the story, for people just to go ahead and die, then there's no story, okay? But if they're taking a risk and then something harms them, then that's a huge story, all right? And that shapes, that is about risk, and the media is much more interested in blame, I think, of the entire community. But in fact, in life, you must take some risks to have some gains, and if you don't, they're trade-offs, but they are more invisible. There's trade-offs.
The media have a very hard job. They got to sell their product. Mm-hmm. And I believe most journalists want to, deep down, give a fair, balanced, careful view. But it's hard to get that on the front page. It's hard to get a lot of viewers. And so there's a tension
when a scientific discovery comes along that's exciting, you know it's scientifically important, but to get people to pay attention to it, it's sometimes necessary to say, it may hold the key to whatever, curing cancer. Otherwise, the readers won't pay attention. And Bert says maybe the scientists are complicit in that too. But we're all complicit in order to get people to pay attention, potentially overclaiming those things. We're complicit also in assigning blame when anything goes wrong, as Janet says. What's really hard is to figure out, how in our highly connected world we can get people to pay attention to what are actually complicated, balanced stories, because they tend not to sell.
Yes, please, Bert.
I think a good example of what you're asking about is fen-phen. Fen-phen was a drug that was initially approved in Europe, I think, for obesity. Now, obesity is an incredibly huge problem, not only in Europe, but in the United States. It's probably responsible for 50% of deaths from all causes, either directly or indirectly. It was discovered that fen-phen, in a very small fraction of cases, causes severe heart disease. And it wasn't long after that the drug was taken off the market. The media had a large role in that, and this relates to exactly what Juan is saying. Is the risk of taking fen-phen counterbalanced by the risk of not taking fen-phen and being obese? And that story never got played in the media. It was only the downside, the negative. And perhaps we're to blame too, because there wasn't a horde of scientists rising up and saying, yes, it does cause rare side effects, but it also keeps a lot of people from dying of cancer and heart disease and many other diseases.
But isn't there another side of the media relationship? Because we're all agreed that people's buy-in to genomics and the promise of genomics is very important. And the media don't just report on drugs, they report on research sometimes. And for instance, there was a huge media interest in the Human Genome Project, and a huge expectation raised that the Human Genome Project would deliver. And it did scientifically, but whether it delivered in practical terms is something you addressed this morning, Eric. So what does that do to the current debate that expectations were raised but not met?
So Adam, I actually went back, because people say this, and reviewed my own statements in the media as to what was going to become available in 10 years. And I said very little. I said almost nothing will be available in 10 years. It takes 10 years from the point that you really understand the mechanism to get a drug approved on the market. We don't understand any mechanisms. This is 30 years stuff.
I'm not sure.
I said this. Others said this. It didn't make much of an impact. So I think it's worth asking empirically, even when you say those things, as people did, if there are multiple messages going on, if this is a message that there's a scream headline in the New York Times as there is human genome sequenced, it's hard to keep people from having unrealistic expectations. I keep wanting to tell the story of the germ theory of disease and the understanding that microorganisms cause disease, and the 75 years between that point and the availability of cheap antibiotics that treated people with infectious diseases in the developed world. 75 years. That's a triumph that could happen. But it's 75 years. And nobody wants, the Congress in the United States doesn't want to know that fact. The newspapers don't want to know that fact. And we have to keep saying it again and again that we are doing this for our children and our children's children, and we shouldn't be apologizing about that. But it's very hard to get anybody to pay attention that would be wonderful if the outcome for cancer were, from the time that the genetic basis of cancer became clear around 1980, to the point that we had the full understanding of the pharmacopoeia was 2050. And from that time forward, cancer was treated differently, we wouldn't be apologizing.
So Eric is blameless in making predictions that don't turn out. But let me just read you all.
This morning I refused to predict anything.
I'll read you all three, one of which was right on the money, and the other two I think are outrageous. 1990, Walter Gilbert, the Nobel laureate who was involved in the development of sequencing, in 1990 he said, in 2020 you will be able to go into the drugstore, have your DNA sequence read in an hour or so, and given back to you on a compact disc so you can analyze it. That was very perceptive at the time. People thought that was nonsense, but it's going to happen. Okay, so that one.
Well, the compact disc will be obsolete.
Oh, that's obsolete. Okay, all right. That will
Wally
give him.
got that bit
We'll
wrong,
give him that.
but-
That's nitpicky.
But remember, he's got a few more years. Now, in 2001, that was about 11 years ago. Francis Collins, by 2020, the impact of genetics on medicine will be widespread. New gene-based designer drugs will be introduced into the market for diabetes mellitus, hypertension, mental illness, and many other conditions. I think that one is a stretch. And then,
very unlikely.
And then this year, Dr. Collins said, we are approaching 1,000 validated genetic variations associated with common disease. So I think we have 1,000 new drug targets sitting in front of us. I think that's a stretch. In fact, that would ruin the pharmaceutical companies if they pursued these 1,000 drug targets. So anyway, the point is, I think scientists are probably as much to blame as the media in certain situations where one does really go out and make statements that just aren't going to pan out.
Well, a more recent example of Eric's infectious, and I joined the FDA at a time in the 80s when people were just totally hyped about monoclonal antibodies, okay? It's taken really 30 years for them to really come into being a foundation of therapeutics. And that may be upsetting and everything, but really it takes a technology, even once it's in the clinic, a long time often to find its way. And hopefully that's better than 75 years. So maybe the next round we can we can compress this time, but still it takes a significant amount of time.
Okay, let's go to some more questions. Is there a question from the online audience? Yeah. Sorry.
Great. We have a question from a med student in Israel. So he's asking, what kind of drugs do you think we're going to have in the future? And also another student from the States asks, is this financially viable for government and healthcare programs with these new drugs?
Okay. Well, we spent quite a long time talking about drugs, but let's get to the question and also tie in the costs, because at the moment we're in a situation of escalating supply of drugs, but of healthcare solutions anyway, and escalating costs, which nobody thinks is going to be affordable in the long term. So who wants to tackle that question? Bert.
It's obviously a difficult question, but I'll answer it, or try to answer it, with respect to cancer. So right now there's targeted therapies. They work to a certain degree, but they're not going to cure cancers. What is going to cure some cancers are some combination of new therapies, immunotherapies, perhaps gene therapies, and mysterotherapies. Now, what's a mysterotherapy? Mysterotherapy is mysterious therapy that has yet to be invented. Now, who is going to invent it? It's going to be people like in this audience, the young people, because only those people don't yet know what won't work. We senior investigators know what won't work, so we don't try it. And I think one of the challenges that was mentioned in this morning's session is to make sure the young scientists have the opportunity not just to do the very next step, the very next targeted drug that has a slight modification and a slightly different tyrosine kinase inhibition profile, but can think more globally, the kind of education and also the courage to try and do something really new. Because if we're going to tackle these problems, we need new things, and the only people that will do that are the young people. So tell your med student that that's what he should work on.
Janet wants to say that.
Yes, I think that people want to know what are the new drugs, but really I think the foundation of medicine is diagnosis. That's where you should start. And being like in the U.S., like we're going to intervene and treat, but really rapid diagnostics, point-of-care diagnostics that could be used all around the world. I think that really is where we need to go in the future. Not only diagnostics, but actually the pathophysiology underlying that, which is really what Eric was talking about. Those are going to be the advances that really lead us to new therapeutics, in my opinion.
Juan, you wanted to say something?
So, look, I mean, there's some really interesting disconnects here, and they're going to lead some big, really interesting things. I mean, when you're talking about some of the problems of disparity of how you have access to stuff or how you manage stuff in third world countries and here, if the basic problem was the access to new science, then that would be a persistent problem. But we're in a really interesting period, right, where the amount of data that Eric's generating out of his massive factory is of such orders of magnitude, and it is publicly available, that you are now moving into an era where what used to be a very data-poor science is now incredibly data-rich. And if the major cost of bringing a medicine to market or designing something using Bert's discoveries were having to set up massive biolabs and massive biosystems and waiting there for 12 years, then you'd have this disconnect. But when the major part of bringing a medicine to market is how long it takes you, whether your IRB is too conservative, whether the pharmaceutical doesn't invest, whether it takes 12 years, et cetera, you're going to have a system where it is possible that you will offshore a big chunk of the pharmaceutical industry to countries that work in a different way, have a different regulatory system, have a different university system, and what you saw with computer chips, with steel, with phones, with everything else, all of a sudden you may see some bio powers, pharma powers, that look very different in 10 years from what it looks like today.
Which isn't necessarily a bad thing if it gets things moving faster.
I'm not arguing it's a bad thing. I'm saying you could see a very large displacement because there are some real disconnects between what we can do and what we're doing.
And don't forget, for example, in India, they've developed portable electrocardiographs that are much less expensive and inexpensive to operate and can operate anywhere and so forth. They've adapted the technology that we have to a different milieu that works very well. And I think that may happen with drug development because drug development is not very good. It's very labor intensive, it's very long, and it's not very modern. And we could look to the developing world to figure out new ways to do this.
Joe, you wanted to say something?
Well, a question to Eric and Janet, anyone else also. Do you think that we're overwhelming the pharmaceutical companies with the enormous amount of data that comes out to the effect that the size of these companies haven't really changed substantially? If anything, they've been downsized in the last 20 years, and yet they're being flooded with enormous amounts of data and knowing how to choose what to work on and so forth and so on. That's an issue.
So
what do
I
you
think
think?
we're diagnosing, we're placing the problem in the wrong place. You could say, oh my God, all of these people out there have access to the internet. They're being like flooded with so much data. Wouldn't it be better off if they just searched their local library instead of having the entire world's data at their disposal? Aren't they going to be brought to their knees? But no, Google has helped organize that data for them, and others have helped organize the data for them. Our problem is not, I just reject this notion that pharma would be better off not knowing the heterogeneity of disease, not knowing the molecular mechanisms. I just think that's nuts. I think our problem is we have done such a great job biologically of continuing to open up the pipeline from the very beginning with the double helix all the way down now to basis of disease, and it's crunching at a bottleneck. And this bottleneck is our problem. And pharma is still operating in the old way of not saying, how do I not react to all this data in the old way of, like Francis Collins's quote, which I don't agree with, 1,000 new things means 1,000 new targets. It doesn't. It means a collection of leads that might point us to 4 pathways, for example. Or it means sift it for the rare families that have some circulating factor. Pharma's not sifting the information. So I don't want to apologize for lots of information. I want to say the next generation has got to get out there and now make very efficient ways to sort through information, and then we have to have economic return to those people who do it.
Tikki, you wanted to make one point.
Yeah, I think this issue of clinical trials moving offshore, that's the reality. The data that we collect indicates that in Russia, India, China, Brazil, South Africa, there's been, on average, about a 200% increase in the number of clinical trials that are now going out in the developing world. Now, cost is one of them. The other one would be the perception that regulatory systems are a bit lax, and of course the third being the large number of potential participants, which brings into focus, unfortunately, that a few of the pharma have been engaging in rather unethical practices when they're doing trials in the developing world. And as you know, there's been a record number of fines. Pfizer got hit with $3 billion fine over a trial of Trovan in Nigeria. So to me, in the future, what's important to go with that outsourcing is increasing transparency and accountability. And the WHO actually 6 years ago established an international clinical trial registry platform where we requested all clinical trials, even phase 1 discovery trials, to be registered with 20 minimum items even before the first participant is enrolled in the trial. So I think that's the key issue, that there are many people in the developing world who are worried about their populations being exploited.
Yes, I suppose it comes back to that idea of popular involvement in what's going on. It's not just being receivers of information, or in this case, receivers of clinical trials, but without the genomic research infrastructure behind it or anything like that. Yeah. Okay, there's somebody who's been waiting very patiently with a microphone, so please, yes.
Question.
No, please use the microphone.
We hear a lot about new drugs, but what the panelists think about the repurposing drugs, like drugs that have been already available, sometimes without a patent anymore, and you can try to use to another disease. So the repurposing of a drug.
Repurposing of drugs off patent. Can we, if we do answer that, could we answer it within the genomic research sort of environment? Because we're really here to talk about genes.
There's one good example of exactly what you were saying that's related to genomics. The current best therapy for colon cancer is aspirin. Far better, it appears, in a preliminary study published about a month ago in the New England Journal by Walt Willett and the Nurses' Health Study, that hardly anyone who takes aspirin who had colon cancer dies from it. But it only works in those who have PIK3CA mutations in their tumor. So that's a gorgeous example of how repurposing of an old drug could be coupled to genomics. And I think there are companies who are thinking about how to do that in the future.
Actually, rapamycin and renal cancer is another one, right? Is that right?
Mm-hmm. Let me take another question from the audience, another patient person.
Hey, I was wondering how big a problem you think publication bias is. AI, pharmaceutical companies doing a lot of small studies on one drug and then selectively publishing only the positive results.
Perhaps that question could be widened to say publication of positive versus negative results generally in science.
Of course.
And how does that affect the delivery of healthcare? Who wants to address that?
Janet. I'm not sure you did any favor by widening it, because a clinical trial is a well-defined entity. And I think it would be a totally reasonable thing to say every clinical trial must be published. Human patients, and you run a clinical trial, there is an ethical obligation when you have used the human in a study to get that data out there so that there cannot be that bias with regard to the human population.
Well, I agree with that, and certainly the EU, EMA, has recently said they're going to publish what the data they receive. Unfortunately, they don't receive a lot of the primary data. The US FDA, for pre-marketing approval, gets all the data. Unfortunately, we can't publish it. So I think in the US, the Congress has dealt with that with clinicaltrials.gov, which is you're required to register all your clinical trials. That doesn't mean, there's also a results section that's supposed to be published. But of course, it isn't in great detail, and it isn't as if people are necessarily checking on that. But at least clinical trials are being opened up more. I agree with Eric. I think that eventually, probably in the not too distant future, there will be some mechanism whereby all clinical data eventually will get into the public domain. Because after all, human beings volunteered to participate in that, and it's kind of like the human genome in some ways. It's kind of hard to believe that somebody owns that, but that isn't the case right now.
Yeah, just quickly, I think what Janet just mentioned is that the registration of clinical trials will hopefully be one way of overcoming the publication bias. And I just remembered reading an analysis in Nature a couple of years ago, which brought up two fairly disturbing observations to me. The first was that trials that have positive results tend to get published much faster than those with inconclusive or negative results. The second one's even more disturbing. If the trial is supported by a pharmaceutical company, it's much more likely to give you a positive result rather than if it was in the academic sector. So it's a definite problem.
Let's go to another question from the online audience. Oh, sorry, you haven't got the microphone. One's coming down. I'm sorry. You could shout from there, really, couldn't you? Okay, good.
Okay, so we've been talking about a lot of bottlenecks, obstacles, barriers. Can we try to focus more on how do we get there? What kind of questions do we have to address? What kind of people do we have to talk to? What should we do?
Lovely, positive question. Right.
Good. I have lots of suggestions.
Okay, I think I'm going to ask you all for suggestions on that one. Go for it.
All right. Well, I believe, number one, that we should develop worldwide, we should develop clinical trial consortia and not do trials for individual agents or whatever, but have the professionals who work in any disease able and ready to evaluate anything that comes down the pipeline. This will remove a large amount of the lead time and that takes to get trials up and running. We're doing a trial in the United States called the I-SPY trial that takes multiple breast cancer drugs and evaluates them in an early phase. So I think that's one thing that should be done. I think in the genomics area, we've talked so much about, okay, we've had the basic sort of reductionist, I call it, with all due respect, okay, the science of understanding structure of DNA, understanding the gene. But we really need to have put more resources, I think, into understanding that middle ground between the human being and the gene, which is only very clear for a few genes, okay? And all the other genes we don't really know that much about. And so and we need, from my standpoint, I'm at the end of the process. So I'm at where you evaluate drugs. I'd like to see more effort be put into developing biomarkers, diagnostic tests, and other things so that while we wait 30 years for all of you, okay, to make the new discovery so that biology is totally understood, okay, all those people who are suffering from diseases in the next 30 years can have better hope. And we are doing in consortia, we're working with IMI in Europe, we're working in the United States with a lot of consortia, public-private partnerships to get a lot of this work done.
Thank you, Janet. Bert.
Yes, this relates to what we were talking about in the first session a bit. The reason that Joe's work was of such monumental importance was not because so many people were affected with the deficiency for the enzyme. It was because it opened up knowledge about a pathway that led to prevention of heart disease. We still today can't cure anyone better who has a massive myocardial infarction than we could when he made his discovery. And Mike, this relates to what you were saying about the value of genomics research in illustrating pathways. But that idea of prevention, we've talked this whole conference, right? It's about curing. And in cancer, that means curing patients who have advanced disease. I think that's Plan B. Plan A should be preventing them from ever getting cancer. And there's very little funding, very little research done on prevention, early diagnosis. I think that's true in other diseases as well. And as a positive thing, I would say if we could somehow convince the research community and the funding bodies of the long-term view that what their mothers say is really true, an ounce of prevention is worth a pound of cure, then we would get a lot of pounds out of that.
Thank you. Who wants to go next? Joe, do you want to say?
Well, I think both Janet and Bert have said it well. I would just add one other thing is that I think the choice of the target and its relationship to the pathogenesis of the disease is absolutely key. And I'll just, two recent examples. I think the discovery that TNF was important in the pathogenesis of rheumatoid arthritis. It seemed to be at the hierarchy of all the cytokines led to anti-TNF therapy. And there are 3 drugs that have been really major drugs for not only for the drug companies, but for the patients that have really changed their lives. And then the other one would be, everybody knows the story about Gleevec. That was a very, very clear target. One developed, the scientists who did this developed the right models in tissue culture first and then in genetically engineered mice. And so I think those are 2 good modern examples of the target chosen were absolutely key to the pathogenesis of the disease. And that would made the trials very simple and straightforward.
Tikki, who should be?
Yeah, how do we actually get there? I mean, my view is that for genomics-based interventions and tools to really reduce this inequity that we're talking about, it has to be introduced in the developing world in the context of the healthcare delivery system. You're talking in many countries about healthcare systems which are fragile and sometimes completely dysfunctional. So introducing something that's new has to be seen within the context of the whole system, not just as an intervention to fix a particular problem.
Thank you. Juan, looks like it falls to you to have the last comment.
So I'm going to do something really stupid. Against the strong and sage advice of Eric, I'm going to make a prediction about the future.
It's on film.
It's on film. I'm sure
Joe's going to quote it back.
I know, and it's going to be ugly.
What was it, year 2012?
So look,
go for it.
There's something out there, you know, some people call Amara's Law, which is almost every technology tends to be overestimated to begin with and underestimated to go forward. And I think we're at a period where we're underestimating what all the folks on this panel have done. I think just the sheer amount of discovery, putting out the genome, the proteome, the metabolome, the epigenome, everything else, it's going to be seen as an absolute breakpoint in human history when you moved into writing life code. And I think we are underestimating actually what all this is going to do. I don't think you'll see it. I mean, if you've got a cancer right now, boy, is it frustrating because you should be farther along. But I think we're in an incredible period, and I think this is going to be a breakpoint in human history. Now, on the specifics of what we can do, you have some very specific examples out there. Most pharmaceuticals don't like vaccines. They don't like vaccines because if I can sell you 1 pill every day for 10 years, or I can give you 1 shot and see you in 10 years, guess which one I'm going to pick? So because of cost and liability, almost every company that made vaccines came that close to going out of business. And there were at one point only 3 or 4 pharmaceuticals left. When you put a cap on liability and you put incentives on vaccines, that makes an enormous difference in production, in pipeline, in market, and in public health. AIDS drugs. When you get angry and you act up, and I'm surprised the cancer community hasn't done this, you get results, and you get results fast. The final thing I'll say is, with all this data out there, I think there's an incredible opportunity for a whole series of countries that have not played in the pharmaceutical game to take these databases, to use their smart programmers, and we've got 1 billion more smart minds coming online, simply because the schools have gotten so much better in Singapore, in China, in India, in parts of the United States, in Canada, in Mexico. And boy, are we able to see some of this stuff. So integrating these global networks and getting those networks faster, and coming back to your point, having cross-approvals. So if it's working over here, let's approve it fast over here. Let's not start clinical trials all over again. I think we could really accelerate the system and improve the quality of life and lower cost.
Thank you. That's a hopeful point to end on. Eric, I didn't give you a chance to comment on this one, but do you want to go?
Sure. I think you make a really important point, and it's in many of the points here. Incentives and infrastructure are so important as to whether we're going to deliver on this in the course of 10 or 15 or 20 years, or it's going to take 50 years. As much science as we can do, and as great as it might ever be, we have to do things that currently work against the model. Sharing information, sharing genomic information across the world will either dramatically accelerate or, if we don't do it, dramatically slow things down. But we're not set up to do it. We're not set up to share. Sharing patients across clinical trials. The incentives aren't aligned to do that right now, but there's an ethical obligation to do so. Standing clinical trials capacity, like Jana talks about, where you can drop your drug into it rather than bringing up a clinical trial, running it, and bringing it down. But that requires people doing unnatural acts, sharing with each other a clinical trials infrastructure and supporting it even when you're not always using it. Working on prevention.
That's a subtle hint, isn't it? Yeah.
Working on prevention. All sorts of things that go against the economic incentives. And I think you've given good examples of it. We have to rise up above it because some of this stuff is going to take common cause that isn't economically incented. But we've reached the point where it is the difference between whether we deliver on this scientific promise or whether we have to explain to our children why we failed.
Thank you. Well, that's a good point to end on. There's a glass of wine outside waiting for you all and us all. And we have a half-hour break before the concluding panel, but I'd like to thank the panelists and all of you, audience.
The old folks here.